Ever wonder how we all have the same features but none of us look the same? The answer is genetic variation. Our DNA is comprised of the genetic information from both parents and is susceptible to changes in the genetic material of a cell or virus resulting in a mutation. Wait a minute, a mutation? That sounds scary, and sometimes it can be, but other times it could be harmless. Once more, a mutation is a change in the genetic material of a cell or virus caused by a mutagen during the cell division process of meiosis or mitosis. Mitosis, you say? It's the process of a single cell dividing into two identical daughter cells. Its purpose is for growth and to replace worn-out cells. The cell in mitosis spends 90% of its life in interphase. During interphase, the cell's DNA undergoes replication, which is oftentimes when the problems start to occur. Cyclins and cyclin-dependent kinases are the regulatory proteins during the cell cycle. They act as checkpoints for each stage of the cycle. Maturation promoting factor, or MPF, is the complex that moves cells from G2 to M. These systems are very effective, however, they do make mistakes. The first type of mutations that can occur is called a point mutation, which can be divided into base pair substitution and base pair insertion or deletion, in which a nitrogen base can be changed, not fitting quite right. An extra nitrogen base could be added or even deleted. Then there are silent mutations, which have no effect on protein synthesis because of redundancy. Missense mutations are changes in the DNA that code for amino acids, but not necessarily the correct amino acid. For example, sickle cell anemia is a missense mutation caused by a mutated version of the hemoglobin beta gene, resulting in abnormally shaped red blood cells that can interrupt the blood flow. Nonsense mutations stop the coding for amino acids, nearly always leading to a non-functional protein. Cystic fibrosis is a nonsense mutation in the CTFR gene so that neither allele can produce a functional CTFR protein. Insertion and deletions of nucleotides completely disrupt the reading of the genetic information, causing a frame shift. A mutation specifically deduced from mitosis is cancer. It is caused by a mutation of genes that regulate cell growth and division, leading to the growth of tumors and metastasis, or the spread of cancer from its original location. While proto-oncogenes are responsible for the normal cell growth and division, a conversion to an oncogene leads to an abnormal stimulation of the cell cycle, resulting in the production of cancer-causing genes. This conversion is either caused by movement of the DNA within the genome, amplification of a proto-oncogene, point mutations in the proto-oncogene, or mutations in the RAS and P53 genes. Mutations in the RAS gene lead to an increased cell division, and P53 genes prevent suppression of the cell cycle, which is exactly why cancer can spread so quickly. But wait, there's more. Mutations in meiosis, you say? For starters, meiosis is the sexual cell division of a diploid cell from two sets of chromosomes to haploid cells with a single set of chromosomes, thereby creating gametes, the sperm or the egg. In anaphase, the homologous chromosomes are separated. However, if they do not separate normally, one gamete will receive two of the same type of chromosome, and another gamete receives no copy. This is called non-disjunction. If non-disjunction has already occurred, aneuploidy is subject to happening. Aneuploidy is a result of the fertilization of gametes that have underwent non-disjunction, causing the offspring to have an abnormal amount of a particular chromosome. There can also be a monosomic zygote. This means that there is only one copy of a particular chromosome. There can also be a trisomic zygote, which has 3 copies of a particular chromosome, and finally there's polyploidy, where the organism will have 2 complete sets of chromosomes. And that pretty much sums up mutations. Pretty simple, right?